I'd like to start by thanking the organizers for putting together this really interesting and important event and to invite me to take part in it. I hope to make justice to the topic of emotions and also to women in science. So today I'm going to be telling you about fear, a fundamental emotion. And what I mean by this is that fear is actually crucial for survival and therefore is present across the animal kingdom. So I'm going to tell you about how we're studying fear in the lab and how one might think about how this generalizes across species. So I'll start with a video where you see a deer drinking water, and you'll see that its environment is going to change very suddenly. Okay, so you see that in a split second, this animal that was faced with a threat changes drastically its behavior. And it's easy for us to imagine that its internal state also changed quite drastically when we project ourselves onto the deer's experience, and we might imagine that he became very alert, that its heart started pounding, and so on and so forth. So one might conceptualize this process, the following way. So you have an object which is a threat. In this case, I'm illustrating this with a bear. And the detection of the threat, this object, is going to trigger a change in the organism's state, body and brain state. And that is the emotional state. And this state is going to promote a response that is hopefully going to mitigate the consequences of this encounter and improve the chances of survival. And in this case, I'm illustrating this as an escape response. Now, I guess this is how most people think about the sequence of events that lead to fear, which is you detect a threat, and then because you detect a threat, you become fearful, and because you're fearful, you'll escape. But one might conceptualize it differently. So, for example, ages ago indeed, William James, already in the late 1800s, proposed that maybe something else was going on, that you would detect the threat, the object, and that was going to trigger a response. And it's actually the response, the perception that I'm escaping, that actually induces the emotion, fear. Okay, so nowadays, actually, different researchers approach the topic of emotion from different perspectives that include both this idea that the emotions serve a purpose of facilitating the way we deal with the environment. In the case of fear, it helps us deal with a threat, but also that emotions are constructs that rely on the appraisal of what's going on around us, the changes that those induce in our body, and how we construe the feeling or the emotion of fear. In this case, if there's a threat in the environment, that my heart starts pounding, I start running, I perceive that, and that's going to induce the emotion. And so you can take all of these approaches to study emotions. In my case, I'm going to talk to you about how the detection of an object is going to induce a change in the organism that is going to facilitate the way it deals with this threat by changing their behavior and internal state. Okay. So to start, I would like to emphasize a number of features that will go through my talk that pertain to these emotional states. So the idea is that an internal state, the emotions are internal states that are triggered by external threats, as was mentioned before by Hasman. An important thing is that it scales with the intensity of the event. So imagine, if the threat level is higher, the emotional state, the fear is going to be stronger. It also is important that it outlasts the event. So, for example, you might imagine that in the encounter of the deer with a crocodile, the deer is going to be in a state of heightened alertness for longer than just the brief encounter with the crocodile. And then finally, that the context has a very strong impact, not only in the state change, but the behavioral responses that you might see. So this means that it's not just the detection, say, of the crocodile that matters, but what's around the animal, whether they have space to escape and stuff like that. So the context has a very strong impact on emotions. Okay. So one important thing about emotions is because, this is moving. Because they are crucial for survival, and they are going to be present across the animal kingdom. And not only they're present, but they also share commonalities. And so what I'm illustrating here is that all visual elements ever tested, they respond to expanding shadows as a threat. And that's because predators, when they approach, they produce an expanding shadow. So most animals will respond to an expanding shadow as being threatened. Now, once they detect a threat, they also show similar kinds of responses. So you have, for example, 3 categories of responses. You have freezing, and the idea of freezing is that it allows the prey avoid detection by the predator. You have flight, so animals can escape, and this is going to increase either the distance from the prey to the predator or the ability to find a shelter. But if neither of these options are available, then you still have the option to fight the predator. And all animals tested show some of these options. Of course, the way they instantiate, say, for example, the escape is going to depend on whether it's an animal in the ocean, in the water, or a bird, or a mammal, or any other species. Okay, but the important point is that because there's commonalities, by studying these processes in one animal, we're probably going to learn about how other animals would deal with the same problem. Okay. All right. What I'm showing you here is the brain of a human and the brain of a rodent. And the point of this slide is to show you that not only are the stimuli that animals respond to or the kinds of response that they produce similar across species, but actually the brain areas involved are also similar, at least across mammals. And maybe more, but I'm going to stick with mammals for now. And so what I'm highlighting here is structures in color are going to be homologous. They have the same origin in human and in the rodent. So for example, here the prefrontal cortex and so on. Actually, my 2 favorite brain regions within these circuitries is the hippocampal formation and the amygdala. These are brain regions that I've devoted a large part of my scientific career to, and that actually I think Liz Phelps is going to tell you a lot about. But they play a very important role, quite distinct. So the amygdala is very important, for example, to the regulation of the fear responses, whereas the hippocampal formation is going to be very important in forming the memories of the event, in this case, the encounter with the predator. Okay. I was interested, as I told you, in these processes, and I started studying rodents and studying the amygdala and the hippocampus. But more recently, I became interested in how distant animals with very different morphologies, very different bodies, very different brains, that occupy a very different niche, solve this problem, this same problem. And how could I learn from these other animals and see to what extent I can push this idea that things are generalized? So for that, I resorted to studying fruit flies. And first and foremost, it's important to know that fruit flies are subjected to also extreme threats. And also, that these are one of the reasons to say I could have studied any small bug, but actually the fruit fly is one of the most important model systems, model organisms in life sciences. And so this has a number of reasons, historical reasons, but also because there's a lot of commonalities across fruit flies and humans, surprisingly so, for many of you maybe. So for example, the fruit fly has about 15,000 genes, and humans have about 22,000, which is not a lot more. It's not like an order of magnitude more. But importantly, 60% of the Drosophila genes, so the fruit fly, have a correspondent in the human genome inherited from a common ancestor. And this is very important because it allows us to study in the fly the function of these genes. And with these studies, we can then learn how these genes affect, say, human physiology, if what we care about is humans. Finally, you might seem that, well, this is a big discrepancy. You have fruit flies have about 200,000 neurons, and humans have about 100,000 million neurons. Okay, so that's a big difference. So one might think, all right, why would you even, as a neuroscientist, study a fly brain that is so drastically different from a human brain if you're motivated by understanding the human brain, which is not necessarily my case, but that's another story. Okay, so, but importantly, actually, there are motifs that appear again and again and again in the animal kingdom that show us that actually to achieve similar functions, you have similar circuits. So what I'm illustrating here is one circuit that allows integration of information. So imagine you have a neuron here that is receiving information about the speed, the size, and the contrast, say, of the threat. okay? And this neuron integrates this information and broadcasts it to downstream neurons. And this is an organization that happens in all sorts of animals that we looked at, from worms to fish to humans. And another motif that I thought would be interesting to illustrate is that if you want to create oscillations, okay, activity that oscillates, for example, to breathe, you need to have oscillatory activity. And the way you do that is you hook up two very distinct kinds of neurons. So in here, in green, you have excitatory neurons. This means that if this neuron is active, it's going to stimulate activity in the downstream neuron. If you have an inhibitory neuron that I am portraying here in red, it does the opposite. Activity in one neuron is going to decrease the activity of the next neuron. And so if you hook up two of these, you're going to create oscillations. And that is, again, something that you find in worms, in fish, in humans. So I hope that I made across the point that there's actually a lot of reasons why one could study the fruit fly to understand how other species might respond to threats. So in my lab, we do this by studying all of these things that I mentioned are key features of emotions. We like to understand what constitutes the internal states that are triggered by external threats. And in humans, we know that there's a lot of internal changes that occur, like, for example, the heart, cardiac activity changes. So we'll look at that, and I'll show you that. But we also want to know how it might scale with the intensity of the threat, why and how these states are maintained in time, and how does the context comes to gate defensive behaviors. Okay, so for this we need to threaten the flies, and we do that the same way you would do with many other species, which is to provide an expanding shadow. Okay, so with the way we do this, we have a fly in a small arena, and there's a big screen, and we just make a looming dark disk approach. Looks like this. Okay, so this is the threat for the fly. And then we look at what kinds of behaviors you might see. And I'll show you a video, hopefully, because this doesn't necessarily always work. Oh, it is working. Okay, so the fly is walking around, and then you'll see the LED turn on. That's when the expanding shadow occurs. Okay, so, and you'll see distinct responses. So this fly stays perfectly still. This fly also. This one kind of escaped. And you see a diversity of responses that include freezing and they include escape. Now, we were particularly interested in freezing, which might be surprising because it seems like it's the least interesting of all behaviors. It's just the fly stops. And so I'll show you, I guess, why we find this interesting. And I feel that this is a video from the BBC that illustrates this very well. So you have the lizard, which is the prey, and you have a snake that moves by as if nothing was there. And you notice that the lizard was very still. Then there's a second snake, and this second snake will start approaching, and it will bump onto the lizard, which of course will mean that it's been detected, at which point it will escape. And you now see the consequence of that. You see how many snakes now go after the lizard. I should tell you that it has a happy ending, and the lizard does escape. But the point is really to show that freezing is very adaptive, and that really allows animals to avoid detection because they stay still. And most predators use movement to detect their prey. Sorry. Okay. So another thing that I wanted to point out, a feature that I believe makes freezing a very interesting study to behave, requires your cooperation. So I'm going to ask you to stand up, and I'm going to ask you to do a particular movement, and then while you're moving, I'm going to ask you to freeze. And when I say freeze, please try to stay as still as possible for as long as I keep talking, not until the entire end of the talk, but for a little while. Okay, so please stand up. All right. And I just want you to do a simple movement that requires a little bit of effort. It is just go like this, okay? All right. Yeah, now freeze. No, freeze. No, freeze. Some people are not freezing, freeze. So don't move. So I want you to stay still for a little bit while I talk. And the point that I want to make is that freezing in psychology and many other disciplines is seen as a passive defensive response, okay, to threats. And that escapes corresponds to a active response to threat. This is very distracting to see everyone freezing, actually. I wasn't expecting that. And this notion of active versus passive coping strategies with threat has led to the society at large to see freezing as a non-response, okay? And this has actually a lot of implications in our society. You can now relax. I hope that you felt that while you were standing still, you were not responding. You were effortfully trying to stay still. So freezing is a very active behavior that involves the suppression of movement, and it involves the activity of many brain regions, and it's energetically costly. Okay, so we're interested in freezing, and we know now that flies freeze. So we wanted to know if in flies, this internal state is similar to that of the internal state that we can see in humans and other mammals. And so we started by looking at an obvious candidate, which is the heart. But maybe for some of you, the fact that flies have a heart is surprising. But hearts are actually a quite old invention. You have hearts actually emerge here in the Bilateria group. So all of these animals, most of them some lost, have hearts of different complexities, but they have hearts. And this is the heart of a fruit fly. And what you'll see is that basically it's very simple. It's a very simple heart. It's a tube along the dorsal part of the body of the fly, and it has 2 rows of cells that contract, and they pump cells and nutrients back and forth in the fly's body. So I'm going to show you how we measure the cardiac activity of flies. We put them under a microscope while they're walking on a ball. So this is the fly walking on the ball. And then you have to think that the fly has like 3 sealed compartments, the abdomen, the thorax, and the head. And what makes communication across them is the heart in part. So you have a heart that connects these 2 compartments and pumps things from the abdomen to the thorax and back. And the way we then look at cardiac activity is that we put a fluorescent molecule in these cells, and we can image the activity of the heart. So this is the heart pumping. And this here that you don't see very well is the fly moving. But you can see how we can distinctly track the movement of the heart of the flies. Okay, so what we found from these studies, we found that flies in the ball, when they're exposed to the expanding shadow, the threat, they escape and they freeze, and that's probably mediated by the muscles in the body of the fly, which we're now measuring their activity. But importantly, like I mentioned, there's a heart. And what we found was actually that the heart, just as in humans, speeds up when there's an escape and slows down when there's freezing. So it seems like you have very different species adopting a similar approach to escape and fear and freezing. And so one of the things that we want to do next is to see how is it, this is all misformatted anyways, how the brain coordinates these behavioral and heart changes. And so we look at the brain, and the flies have a brain, and they have this ventral nerve cord, which is sort of the equivalent of our spinal cord. So we want to look at neurons that send information from the brain to the ventral nerve cord, which controls the muscles, the heart, and so on. And so this is just an example. I don't know how I'm in time. I should know. So this is an example of a fly brain. So these are 5 fly brains, the brain and the ventral nerve cord. And in green, you have the neurons. And this is just to show you that we have genetic access to neurons in the flies. And we can use that genetic access to manipulate their neuronal activity and measure their neuronal activity. And this is what we're doing to try to figure out how these responses are coordinated. Okay, but we wanted to see whether in flies, these internal states also scale with the level of threat. So this is just, we've done a simple experiment where you can see what we've done was we manipulated the level of threat. And what we've done was to have an expanding shadow that moves fast and an expanding shadow that moves slowly. And what you can see in this graph is the fraction of flies that freeze. And here is the time of the experiment. Here is the first 5 minutes. We don't provide any kind of stimulus. The flies are walking around. And for each dashed line, is an expanding shadow presentation. And you can see that the flies exposed to the faster shadow are going to freeze more than the flies that are exposed to the slow expanding shadow. So this tells us that indeed in flies it's also scaling the strength of the response and the level of threat. We wanted to know whether the state outlasts the event. And so what we've done was a similar experiment, but now we stop providing the looming shadows. So basically we have the flies walking around, we provide 5 expanding shadows, and then we stop and we see how flies react. And you can see that still by 5 minutes after the beginning of the threat, you still have a substantial fraction of flies, about 30%, that are still freezing. So flies freeze for minutes on end after the threat. So this is an outlasting thing. And this is, I'm almost going to finish. I'm on red. So what does that mean? Okay, so we also wanted to show that it depends on the context. And so we know, and particularly women, if you walk at night alone, you oftentimes feel threatened. But if you are accompanied and there's people around, now I got distracted because of the time, because I thought it was going to be there, not here. They are accompanied, then you feel less threatened. And that's also true in many animals, that there's what's called safety in numbers. So we wanted to see if that was true in flies. And so we basically have flies, a group of flies walking around, and we expose them to the threat. And you can see here that if you have a single fly, there's more freezing than if you have 5 or if you have 10, showing again that flies modulate their defensive behavior by the social environment. So I just wanted to finish by stating that indeed there's internal states that are triggered by external events, that they scale with the intensity of the event. The state outlasts the event, and it depends on context. And I just wanted to end with a small consideration, which is we started talking about how the object triggers an emotion that facilitates a response, but we think this is actually part of a cycle. And the response itself can feed back onto the emotional state. And then also, because you now change the emotional state, you're going to again affect the response. And finally, you have a new emotion. And actually, the emotion can change the object, because if you're now far from the threat, the object itself has changed. So this is a very complex loop. And I apologize for having taken too long. This is the people that I wanted to thank, and I'm going to stop here.
Thank you, Marta.
Sure.
Now we have some time for a couple of questions. So meanwhile, maybe you think about some questions. I remind you that there is a number you can send your questions to, and also the Facebook page and the YouTube page. But I think we already have a question from the online system. So Razvan, you have it?
Thank you, Marta, thank you for the interesting talk. So in your model, there seems to be object, emotion, response.
Mm-hmm.
And a question that was posted online asks, what about the fear of the unknown, where the object is unknown, or there is no object?
So that's a difficult one. So I was starting by the definition that the emotional state was going to be triggered by an external event, and that by definition you detect the external event. The other question, I feel, if you think about this, you're always exposed to external events. So this is a bit of a, so the question then is, do you ever experience a neutral event? Is there something as neutrality? Do you go through life neutral, and all of a sudden there's a change, and now you are in an emotional state? And that, I think, is a difficult question to answer. My feeling is that for extreme emotions, at least, you have like a qualitative change, but maybe this is a sort of a gradient where you fluctuate. But then I feel that at some point you're in a different landscape.
Thank
Okay.
you. We can also take questions from the audience.
There's maybe one question. Do we have a microphone? Yeah, can you have a microphone over there? There is a question from the public. Where's at the very top.
Good evening. I have a question. why do the animals tend to freeze more when the looming shadow is quicker?
That's a good question. So we haven't fully explored this space yet for the flies, okay? There's actually a study in mice that would suggest that the more important feature to decide whether you should escape or freeze is whether you have a looming stimulus or a translational. So the idea being that if it's translational, you haven't been detected yet, and then once you're detected, it looms on you, okay? And that then, like mice, switch from freezing to an escape. So they would tend to freeze more with a translational than to a looming stimulus. Now, the thing is that here, we're doing repeated looming stimulations if you have like multiple attempts from the predator to get at you. And so actually, at the beginning, they don't freeze as much. And I think in terms of the speed, I feel that it has more to do with you have more flies responding, whether it's an escape or freezing, but we haven't looked at that carefully yet.
Thank you, Marta. So now, due to the time, we have to move on. But you will find Marta